There we go. Okay, so the plan for today is to wrap up the rest of this cell biology PowerPoint, get through our last couple of topics here. We're just going to be going over the structure of the nucleus and structure of the DNA within our nucleus. We'll address the process of protein synthesis, and then just look at the cell cycle. So how cells replicate, or the steps that they go through, the stages they go through as they prepare for and actually replicate. So just a few topics to address here. And then I have a little activity, I guess you could call it that, with some example questions that you would typically see like in an exam. So to give you an idea of how to address this content, or what kind of preparations to make, what kind of content you should be prepared to answer about. So yeah, that's generally the plan for today. So before I get into starting to talk about the nucleus, did you guys have any questions or concerns that have come up since I saw you yesterday? Okay. Of course, as always, the more of you that are willing to have your camera on, the more of an interactive environment this feels for me. And then when we get into the group work part, it is a requirement in order to get the credit for doing the in-class assignment that you guys are on camera in your breakout rooms so that you guys can see each other and aren't just talking to a bunch of boxes. So, at least when we get to that point, please have your camera on. And thank you to those of you that are starting to. Okay, so let's get into it. The nucleus. So the nucleus is a really critical organelle in the cell. It essentially are referred to as the brain of the cell in some contexts, because this is the organelle, the structure that is where all of our genetic material, all of our DNA is housed. And at least in a eukaryotic organism like ourselves, where we have specialized organelles, all of our DNA stays protected within this nucleus. The only time it will leave the nucleus is during replication. Okay? And that DNA, all of our genetic material, acts as instructions for creating proteins. And so the proteins are what are actually carrying out all of our genes. So any characteristic that you might correlate with a gene or DNA is going to be produced by the protein that DNA codes for. And so the structure of the nucleus is a bit unique to all of our other organelles because it is so critical, right? It's protecting that important DNA. So we have a special name for the membrane surrounding the nucleus. It is referred to as the nuclear envelope. It is even more selective about what is able to simply diffuse across this membrane, and has specialized pores, these little channels, the nuclear pores here, that control everything that is going to enter or leave. So these are pores with a bunch of proteins embedded all the way around them. Again, controlling the ions that are coming in and out, controlling the proteins that are going in and out, and then lastly, controlling the RNA that is going to enter or leave our DNA, or leave our nucleus. So we have another unique little structure within our nucleus. So the most dense part of the nucleus with the most densely packed DNA in it is our nucleolus. And the DNA within the nucleolus is specifically responsible for creating ribosomal RNA. And ribosomal RNA is what will make our ribosomes. So although we refer to ribosomes as their own organelle, they are not a membrane-bound structure. They are actually made up of nucleic acids. They are made up of RNA. And so the specific RNA that makes these ribosomes is ribosomal RNA. That's why we have that lowercase r there. And again, the nucleolus is responsible for synthesizing ribosomes and synthesizing that ribosomal RNA. So here's an electron microscope image of a typical nucleus. Again, that nucleolus is the densest part, so it shows up with the darkest coloration when looking at it under a microscope. And you can see the openings for all of our nuclear pores here throughout our nuclear envelope. So here's a closer look at that nuclear pore. Okay. And so the nuclear envelope, since it has its own name, we also have its own term for the space in between our bilayer. So the space in between the bilayer of our nuclear envelope is our perinuclear space. Peri is a prefix meaning between. So the perinuclear space is the space between the bilayer of our nuclear pore here. And here's just another electron microscope image showing the same thing. All right. So within our nucleus is all of our genetic material, as I mentioned before. And so our DNA is going to constantly be transforming between a few different stages of how it is packaged, depending on when we need to express a gene and thus need to be able to unravel it to use it to create an mRNA transcript. It's going to change during replication and become even more condensed, or it will, decondense first so that we can make a copy of it, and then become really condensed so that we can keep all the specific genes together with their individual chromosomes before we separate them into two new cells. And so we'll talk about the terms and all the things that go into making up these different structures. So typically in a non-dividing cell, most of your DNA is just organized into chromatin. And so chromatin is loosely packed chromosomal DNA. So some of the DNA will be racked up into these nucleosomes. So DNA by itself are these two interconnected anti-parallel strands of nucleic acids, of nucleotides. That's what gives us this helix that we are used to seeing when thinking about DNA. But it is there are not many times throughout the DNA's life that it is in this actual format. It only unravels into this double helix when we need to get in there to express a gene, or again, when we need to replicate the DNA for cell replication. So most of our DNA is going to spend its time wrapped up in these nucleosomes. And a nucleosome is the term for our strands of DNA wrapped around histones. These are histone proteins. And so that just kind of protects the DNA together and gives us proteins to interact with in order to bring us into this supercoiled, or at least more of a coil that is going to make up our actual chromosomes or chromatids. And so again, this would be for replication. This is really compacted DNA. You can't get in there to express the genes when it is wrapped around these histones. They are protecting it. And so this is really just to keep it all together during replication. Or if there are genes that are permanently inactivated, which all of us have, we do have genes that are almost never expressed. They will be in this nucleosome structure organized into chromatin for most of their life, right? It will only be unraveled if we need to express a gene or if we need to replicate the DNA. So during replication, or we have slides on replication, so we will come back to this idea. But this is showing the typical structure of how our replicated chromosomes are going to be organized in order to replicate the cell. So we have 23 pairs of chromosomes, so 46 chromosomes total. They're in pairs because you get one of each pair, or one copy of each pair from each parent, right? You got 23 chromosomes from your father, 23 chromosomes from your mother, and those form homologous chromosomes. Let me see if I can type in here. Let's insert a text box. I'm gonna do some terminology here. Those pairs of chromosomes, so you got one from your father, one from your mother, those form homologous chromosomes. And homologous chromosomes contain the same genes, and so they code for the same stuff. Like you would get one gene, or one of the chromosomes from your father would have the gene for eye color, one of the chromosomes from your mother would also have the gene for eye color. And then whichever gene is expressed is based on other factors like dominance and genetic interaction. But your homologous chromosomes carry those same genes, but they will almost always have different DNA because you got them from different sources. And so these are what make our chromosome pairs. And I'm identifying this or defining this to not be confused with what a sister chromatid is. So we have homologous chromosomes, which are always present in your nucleus, but then our sister chromatids are only created for replication. So sister chromatids are the term for the exact copies of each chromosome that are made before the cell replicates. So in a sister chromatid, you would have the exact same genes and the exact same DNA, because these are going to be split apart. One of each chromatid will go to each new cell, giving those new cells a copy of every single gene that you started with. So sister chromatids contain the same genes, same DNA, because they are exact copies of the original chromosome. Exact copies of one original chromosome. But these are only present during replication. So these homologous chromosomes are always present in the nucleus, and then sister chromatids are only present during replication. These are the exact copies that were made so that we can create two new cells with the same set of DNA that we started with. Okay. Sister chromatids are connected through proteins called kinetochores, and two kinetochores creating that connection is referred to as the centromere. And so the microtubules that come out as spindle fibers are going to attach to the kinetochore of each sister chromatid. And so you have a spindle fiber coming from both sides, and then they will pull each sister chromatid away from each other towards the centrioles, those microtubule structures we introduced earlier. The cytoskeleton, these centrioles, right? So these centrioles act as anchors. They will send out other microtubules, spindle fibers, and those spindle fibers will attach to the kinetochores and then pull each sister chromatid back to a different centriole on opposite sides of the cell. And we have slides on the stages of cell replication, so you get to see some of that depicted more in depth. But these two terms get confused all the time, so I just wanted to spend some time talking about it. All right. We talked about histone proteins used to wrap up our DNA into these nucleosomes. And then the term for these loosely packed DNA within our nucleus, when it is not dividing, is chromatin. Chromatin is the term for all of your chromosomes, all of your DNA that is in the nucleus. All right. Are there any questions or concerns about the structure of the nucleus or the structures of chromosomes and DNA that we just discussed? Okay. Okay. So next we'll look at protein synthesis. So this is the core of how everything gets done for all living things.
Sorry, Professor.
Yeah, go ahead.
Okay, so one of the quiz questions had, what is a nucleosome?
Mm-hmm.
Is it like protects the amino? I was listening to what you were saying, but I didn't really see that on the answer. Because you said protein, which is amino acid, no? Am I wrong?
Proteins are composed of amino acids, yes. So the histone proteins that make up the nucleosomes themselves are made of amino acids. But the function of the nucleosome itself is to protect the DNA. So these are nucleic acids. Yeah. So the DNA wrapped around these proteins, which just keeps them protected, so they're not exposed to any potential damaging proteins or damaging ions or anything else that might mess up the sequence of our DNA.
Okay, thank you.
Very cool. And it helps them form our chromosomes and chromatids. Okay, so protein synthesis. As I mentioned earlier, DNA provides the instructions for how to create these proteins. And so the process of protein synthesis is then carrying out those instructions. And this is the core of how all living things express their genes. And so it is often referred to as the central dogma of biology. And it is the core of everything else that we study in order to understand how living things work. And so we have two processes as a part of protein synthesis. Those processes are transcription and translation. Transcription is describing our taking a segment of DNA, so taking a gene, and making a copy of it, a transcript, out of mRNA, messenger RNA. Then that messenger RNA can leave the nucleus so that our DNA never has to. DNA stays in the nucleus and stays protected. That mRNA transcript will then leave the nucleus and take the transcript of that gene to a ribosome, and the ribosome will translate that information into a string of amino acids, so into a polypeptide. So transcription is making that mRNA transcript, the copy of the gene, and translation is translating that information from the transcript into an actual functional protein. So we're going to take a close look at all of the stages here. You're not going to be assessed on being able to determine what an amino acid sequence would look like from a given gene. That is something that I might do in a Bio1 course if we had an entire week to talk about transcription and translation. So most of this, what we're talking about on the slides, is just FYI review, but making you familiar with, again, the structure of DNA itself and then how that actually turns into an amino acid sequence. So we talked about nucleic acids and the different types of nucleotides in our last chapter. And so essentially within DNA, we have 4 nucleotides. Or can I make this smaller without messing stuff up? Yeah. So within DNA, let's just lock that in and get another text box. So for DNA, we have 4 nucleotides, A, T, C, and G. And then for RNA, we just have 1 nucleotide difference. Instead of a T, they have U for uracil, but C and G as well. And we have this double-stranded formation here where we have very specific bonding between nucleotides on each strand. And so essentially, even though we have 2 strands of DNA, only one of those strands is actually going to be read for the mRNA transcript. So that's the one that's carrying the correct nucleotide sequence to code for the protein that the gene needs. The other strand is just there to protect it, essentially. And so we'll have kind of a nonsense sequence, and that is our complementary strand, or coding strand, because it will be essentially the same as what the RNA transcript comes out to. So within these nucleotides, let's see, A will always bond with T, and C will always bond with G. So we always get a purine bonding with a pyrimidine, and it is specific in this way. So if you had 1 strand of DNA, 1 sequence of nucleotides, you would be able to determine what the opposite strand, what the complementary strand would look like, because everywhere you see an A, there's going to be a T on the opposite strand. Everywhere you see a T, there's going to be an A on the opposite strand. And same goes for cytosine and guanine. So for RNA, when we're making a transcript, instead of A bonding with T, A is going to bond with a U. They do not have thymine, they have uracil. And so it's just that slight difference. And so when we are creating an mRNA transcript, in order to make our complementary codon or add our complementary nucleotide, mRNA is going to add a U everywhere that there's an A here, instead of a T. Okay. Hopefully that makes sense. This is just the general concepts that you should recognize at all times about DNA, or should be aware of for moving forward in biology in general. Adenine and thymine bond, cytosine and guanine bond, but then in RNA you get uracil instead. Okay. So the process of transcription requires us to kind of unzip, or to literally unzip, this double strand of DNA so that we can get access to our template strand, which carries the actual gene. And so we have a particular protein here called RNA polymerase, which is going to bond with our template strand at a very specific region called the promoter. And then it'll start to move down the template strand, and one at a time, it'll start building this mRNA transcript. It'll just add the complementary nucleotide until we have a long strand of mRNA, which codes for the specific gene that we were just reading. So this transcript that we have just made of mRNA will then leave the nucleus. It will travel to a ribosome, and that ribosome will then start to translate our mRNA transcript into an actual protein. And so I have a slide for each of those stages there. And so another core core concept within transcription and translation is the idea of codons. Does anyone remember what codons are and why they're important here?
Like the three that are together, I think.
Yeah, so it's sets of three. Codons show up in triplets. We have sets of three nucleotides. And what does each of those three nucleotides code for together as one codon?
An amino acid.
Yeah, for one amino acid. Okay, so essentially every three nucleotides in our sequence here is going to produce one new amino acid being added to our polypeptide chain, to our new protein that we're creating. And so within the ribosome is going to start to move down our mRNA transcript. And one at a time, it's going to bring a tRNA or transfer RNA. So we've had lots of different RNA molecules introduced so far. We have rRNA, which is ribosomal, which is what makes up our ribosomes. We have mRNA, meaning messenger. That's what makes our transcripts. And now we have tRNA, transfer RNA, which is what is carrying our amino acids. And so each tRNA molecule has a specific anticodon, which will align with whatever codon codes for that amino acid. And so essentially one codon at a time, the ribosome is going to bring in a tRNA molecule. It will then bond each of these amino acids that are starting to build our protein here. Here's our bond, forming a peptide bond. And then it will release that empty tRNA and bring in another one. So it is constantly shuffling through this process of moving our mRNA transcript one codon at a time, bringing a tRNA molecule that can bond with that transcript, and that will bring the specific amino acid that the transcript is coding for, forms a peptide bond, and then releases the empty tRNA. And so by the end of this process, we've created a chain of amino acids, and each amino acid is bonded through that peptide bond. And so it will be released in its primary form, or primary structure. Right? We talked about the 4 stages of protein structure: primary, secondary, tertiary, and quaternary. When it is leaving the ribosome at the end of translation, it is in its primary structure. So just a string of amino acids. And then those amino acids will form interactions with each other, make hydrogen bonds based on their charges, and that gives us our secondary structure. But then it will take other proteins, such as those that are in the Golgi complex, to help fold that protein into its actual functional form, its mature form, which will be the tertiary structure or quaternary structure. And so that is transcription and translation in a nutshell. That is how all of our cells express our genes, how all living things express their genes and actually live. And so I added this amino acid codon table, not because I want you to memorize this or to recognize it. Again, I'm not going to ask you to take a segment of DNA or a segment of RNA and turn it into a polypeptide, but just wanted to show this as an example of how the codons align with each amino acid. Okay? We can have multiple codons for each amino acid. But yeah, so anyways, there are 20 amino acids that we can use as living things. 9 of those are essential amino acids, meaning our cells can't make them on our own. We must consume them. We must get them from our food. But essentially, they're added one at a time to make a protein using those codons. That's what I wanted to show there. There's a quick video showing animations of transcription and translation. If you would like further help with that, or if it works better for you to see these things happen, which was always better for me, highly recommend you check out those videos. So is there anything I can clarify about our process of protein synthesis here?
Sir, do we only need to know transcription and translation in eukaryotic cells, or do we need to know them for prokaryotic cells as well?
Just for eukaryotic cells for this context, yeah. Yeah. And like I said, I'm not going to get into the specifics of having you transcribe or translate a set of DNA. So just generally recognizing how the process of transcription works and generally recognizing how the process of translation works is all I'm asking for there. All right. So our last function to talk about here is cell division or cell replication. So making two cells out of one. And so cell replication is through the process of what is referred to as the cell cycle. And we have a few distinct phases throughout the cell cycle. Essentially, we have interphase, which is preparing for the actual division into two new cells. And then we have our M phase, which could stand for mitosis or meiosis, depending on which type of replication we're looking at, where the cell is actually going to start separating its sister chromatids and forming two new cells. So all of interphase is just preparation. Okay? And so we have G1 and G2, which stand for growth 1, growth 2, or gap 1 and gap 2, depending on what you're looking at. And then we have our S phase, which is for synthesis, because during S phase is where we are going to synthesize all of our new DNA. So essentially for preparation here, our first growth phase is going to be preparing the ATP energy, preparing the new nucleic acids that we're going to be needing, and preparing any of the proteins that are going to play a role in the synthesis phase. The cell is also going to start growing a bit since one cell is about to become two. We need to produce space for all those structures. The other organelles within the cell, so outside of the nucleus, outside of our DNA, the other organelles of the cell are going to start duplicating during G1, including the mitochondria. The mitochondria is unique because its replication is solely controlled by itself. The mitochondria have their own DNA and regulate their own processes, which is pretty cool. But the rest of the organelles are going to be controlled by the DNA in the nucleus during this phase. So growth 1 was prepping for S. We've prepped our energy, we've prepped our nucleic acid molecules, we've grown in size a little bit. And now during S phase, all of our DNA is going to be replicated. So we make exact copies of all of our DNA strands. Essentially, I can skip ahead here. Essentially, our double-stranded DNA molecule unzips again, the same way that we saw for transcription. It unzips into two individual strands, and then new complementary copies are made all the way down. So if we just pulled like a zipper apart and then made a new zipper for each of the pieces that we just pulled apart, that is essentially how DNA replication occurs. So two of the original strands, or each of the original strands, is used to make a new
Good, how are you?
complementary strand. So we get two copies of the original. Right. So after S phase, we now have duplicated all of our DNA. We have all of our sister chromatids. We're starting to package up that DNA into those histones and then titer into that chromatid structure that I was showing earlier. And then during our second growth phase here, we're preparing all the proteins and energy that are going to be needed for the rest of duplication here. So for the mitotic phase or M phase. And then the M phase is where we actually get the separation of chromatids and starting to form our brand new cells. So similar to what I was talking about with transcription and translation, I'm not asking you to memorize the individual proteins and necessarily the full process here, but you should recognize that the original double-stranded helix is being unzipped, and one of the original strands is going to be used to make each of our new copies. So it gets unzipped, and each strand is used to make a new one. And so by the end of the process, we now have two exact copies of what we started with. And those, again, are referred to as sister chromatids. Oh yeah, see, I did add the same stuff we just talked about before. So here's that definition of homologous chromosomes and sister chromatids, again, to just be specific about those differences. Okay, so we've doubled all of our DNA. We've prepped all the things we need to actually replicate now, and then we can get into the M phase, or the mitotic phase. So that was all part of interphase. This is now the M phase. And our M phase has a few of its own stages and separations based on what is happening during each stage. So we'll talk about one at a time. So at the beginning of the mitosis, it is referred to as prophase. During prophase, our centrioles, those microtubule structures, are starting to produce those spindle fibers, again, those stretches of microtubules that are going to help us separate sister chromatids. And these spindle fibers are going to help push the centrioles to opposite sides of the cell to eventually act as anchors. And so also during prophase, our nucleus is going to start to come apart, essentially. The nuclear envelope is going to unravel so that all of our chromosomes are going to be accessible by these spindle fibers. And so those spindle fibers will start to attach to the kinetochores, holding those sister chromatids together. So this is all prophase. The next stage is metaphase because of the metaphase plate. And so during metaphase, every single sister chromatid now has a fiber attached to it, and they have been aligned along the center of the cell, or generally the center of the cell. They end up in this linear formation, like you can see here. So the very dark colored portions here are the actual DNA, the chromatids, and then the kind of purple spindles are those spindle fibers. They align on the center. This allows for other proteins to come in and essentially check that everything is functioning correctly, check that all the sister chromatids have a spindle fiber attached to their kinetochore before they actually get pulled apart. So ensuring that both new cells will get one copy of the original DNA. Anaphase, our third stage here, is the actual separation of those chromatids. So these spindle fibers will start to shorten, and this pulls apart those kinetochores, so separates each sister chromatid, and will start to bring them to each side of the cell. So we now have one copy of the original set of DNA at each centriole, so at each anchor on the edge of our cells here. And then the last stage of M phase is telophase. Telophase is where we're starting to actually separate our two, what are going to be our new cells. Okay? So the membranes of the cells start to pinch in, just like what we saw with endocytosis and exocytosis, right? The membranes will come together until they meet, allowing them to separate from each other and pinch off, which happens during cytokinesis. So telophase is just the beginning of that separation, creating what is called the cleavage furrow. So the membranes are pinching in to the center of the cell. And then cytokinesis, or cell movement, if you broke this word apart, cytokinesis is where we will actually, those membranes will finally touch and separate as two completely new cells. And that is the process of cellular replication. And that is how all of our cells become two new cells. Okay? And so I do have a table here talking about the various growth factors, hormones that control and affect the cell cycle. There's a lot of regulation, of course, that goes into the cell cycle to make sure things are functioning correctly. But I'm not requiring you to know those very specifics. Again, for these last few processes we talked about, just generally recognizing how they function is really all I'm looking for. Okay, any questions about cell division here? All right, cool. We did it. We got through chapter 3. Like I said at the beginning of class, for the rest of the time here, I'm gonna put you guys into breakout rooms, so into groups. And I'm gonna have you guys work on this little activity that I created. So let me move this over and then I'll share it with you guys. So I'll give you the link to this in a second. Let's just share my whole screen. What I've put together here is just a slide deck and each slide has an individual question, showing you the types of questions that you might see on an exam. I've mentioned this a couple times in class, but I am very partial to open-ended questions rather than multiple choice or very specific factual questions, because I think it gives you a better opportunity to display an understanding and is a much more accurate representation of your understanding rather than looking at which multiple choice answer you chose and if that multiple choice answer was a random guess or not. So anyways, these are mostly, or should all be, open-ended questions that are very similar to what you might see on an exam about this content. So I'm going to put you into breakout rooms. Each breakout room is going to be responsible for a specific set of questions. So we have 13 students here. Let's do 4 groups of 3. So it'll be one group of 4. So we'll have 4 groups. I have 13 questions as well. Wait, 13 questions and 13 students is too perfect. I feel like I should do this individually, but I want group interaction. I'm gonna do the group thing. That's what I decided first. Okay, I'm gonna put you into 4 breakout rooms, so 4 different groups. Each group is responsible for 3 questions, essentially. And whichever group ends up with 4 students, you will be responsible for 4 questions. So essentially, I'm gonna make group 1 the 4-person group. So group 1 is gonna be responsible for questions 1 through 4. I'll put this in the chat too. Group 1 equals Q1 through 4. Group 2 equals Q5 through 8. 5, 6, 7. No, we need 3 of them. 5 through 7. Q3. Whoops. Q8 through 10. And then group 4 to 11 through 13. Okay. And the group number here is going to correlate with your breakout room number. So you should be able to see your breakout room number when you get the invitation. This will be worth, I believe I said 10 points. I'll double check the syllabus, but this is an in-class assignment. You'll get credit essentially for working on this. I'm going to have you guys work on your set of questions in your breakout rooms. Depending on how much time that takes, I may do some rotation of group members to talk about the answers you guys came up with other group members who didn't work on that question. Or we'll just come back into the main room and talk about all of them together after you've worked on your specific set. Okay, so you're about to get an invite into a breakout room. Your breakout room number corresponds with your group number for these question numbers here. I will rotate through to check in on you guys and answer any questions. You should also be able to request help from your breakout room. And yeah, is everyone clear on the instructions and how this is going to progress?
Where are the questions going to be?
On this, right? I need to give you access to the document. Thank you. So I actually have it in Canvas ready to go. I just need to publish it. So you can either go into module 3 of our, is it not here anymore? Okay, I'm just gonna post it in the chat here. So before you go into your breakout room, please follow this link. Should allow you to edit your slides. You can go ahead and click that to open it up. So I'll give a quick second here for you guys to follow that link. And then I'm gonna make these breakout rooms. So I'm gonna stop sharing. Let's create some breakout rooms. So I can make 4, assign automatically, cool. And group 1 has 4 students in it. So again, I'll rotate through every so often to check in on you guys. You can also request help if you need me and I haven't been there for a while. But if you have any other questions, just let me know. You should get your invitations right now. And to resume recording. Speaking of recording, I can either record my own video lecture of me going through these questions and giving examples of like extra info that I would add or any misconceptions that seem to be present on there. Or I can carve out like 10 to 20 minutes at the beginning of one of our future lectures next week, where we can just quickly go over this together as a class. And I guess asking that way. Yeah, so I guess you can just type in the chat which option you prefer. If most people just want me to record, that's all I'm going to do. We'll spend class time on our next chapter content. But if you guys would like to talk about it as a class, with the opportunity to ask questions in real time, I can do that in class. So just give me that feedback in the chat. Take a look at some of the other questions that were on different slides from other groups. And if you have any questions about them or any concerns about them, take this time that we still have in class here to talk about it. But other than that, you guys are free to go if you would like to. I'll stick around for as long as you need me to for any questions or concerns or comments from any of you. But if you're going to leave, then I will see you not next Monday. Monday is Martin Luther King Jr. Day. There's no school. So I will see you next Tuesday to begin lecture on chapter 4, tissues. Yes.